Okay, we're into chapter 4.5. This says membranes and transport, which it is about, but we're going to talk about some very basic chemical principles, particularly this one called osmosis, which is a type of diffusion. These are super critical to understand. We'll talk about the structure of the membrane. So remember, the membrane, cell membrane is the part that surrounds the cell. We'll talk about how this membrane is what we call selectively permeable. That means it allows some things to come and go while keeping others out or in. Then we'll talk about how membranes can form compartments in cells and control the movement of the cell's molecules. This section is very important. Diffusion, osmosis, very important principles to understand. At its core, right, a cell is defined by the cell membrane. That creates an internal environment inside the cell that is distinct or different from the extracellular or outside environment. So this is what defines a cell. And these cell membranes are made up of a combination of lipids, these are the little molecules here, and big proteins, the little yellow things that stick between them. The membrane might be called also a plasma membrane or a cytoplasmic membrane. It's because on the inside here, we have the cytoplasm. It's kind of a fluid that is inside of the cell. It's mainly water, but there's some other stuff in there as well. Okay, so we have the membrane here. We'll talk about its organization in a bit, but it is important to know that these little bits here with the little head and the tails, these are what we call phospholipids. They're a very special type of lipid that will kind of form this shape no matter what. And so why do I talk to you about membranes? Well, the membrane defines the cell. If you have a hole in your membrane, that is bad news because things from inside start to leak out and outside leak in, and usually the cell will die. And of course, we take advantage of this. There are antibiotics that will actually poke holes in bacterial membranes. So polymyxin B, an antibiotic found in like Neosporin ointment, will actually cause holes to be punched in the bacterial membrane, causing it to degrade, basically. So bacteria, where it can punch a hole, they will basically shrivel and leak and die. Some bacteria, of course, are resistant to that. We'll talk about that later in the course, though. So, understanding membranes, right, like they're critical to life. Another reason, some of the proteins that are in membranes are critical for signaling, right? Your cells have to talk to each other, right? This cell has to say, okay, I'm going to be a muscle cell. You next to me, you're going to be a muscle cell. Maybe you next to me, you're like a blood vessel, like a vein or something like that. They have to talk to each other and send signals. But many pathogens can use this signaling and trick our cells. So in this case, HIV virus, can bind to a cell receptor, our protein, on the surface of our T cells. These are immune cells. It binds and it tricks it. It says, hey, I'm not an HIV virus. I actually need to be inside the cell because I'm important, right? So it tricks the cell into letting it in. And once it's in, it does its viral thing. It actually integrates with our genome and it's with you forever at that point. And then it can get out. But it's got to get in. And it does that by tricking the cell through these proteins. And then it gets out through the membrane. So these membranes are critical, right? They have important roles, keeping things out, letting things in, and they can be tricked by pathogens. So as I said, the membrane itself, right? Most of it is formed up of lipids. And these lipids are special ones. They're called phospholipids. That's because they have a phosphate group on the head part of it, and then they have these fatty acid tails. Now, this makes them what we call amphipathic. Amphi meaning both or two. This means that the head part of it here, the phosphate part, is polar. That means it's hydrophilic and it likes water. That's great because outside the cell is generally water and inside the cell is water. But then these tails are non-polar. They hate water. They're hydrophobic. They try to hide away from it. Okay, so these two tails are trying to hide from the water, and that causes them to associate like this. The heads are going to arrange themselves out towards the water, and the tails are going to arrange themselves inwards away from the water. This means that your cell membrane can actually self-arrange itself. It forms what we call a phospholipid bilayer. There's two of them, so that's bilayer. It forms this, and it's super stable that way because of the chemistry here. And it will stay like that. And it's not just the phospholipids. There's other bits in there. Things like cholesterol. You might think cholesterol is bad for you, but it's actually a major membrane component. Of course, having too much of it, it then doesn't go into the membrane and clogs up your arteries. You do need a little bit of cholesterol because it makes your membrane more fluid. It helps it flow and move. Your cells aren't rigid, right? That's why if you poke yourself, you're not hard and firm like a woody plant or something like that. And actually, the arrangement of these tails, remember I talked about saturated versus unsaturated. If the tails have little kinks in them, the membranes are even more fluid. And this is interesting because this makes them flow much easier. And we see that like marine animals that live in very cold water, like in the Arctic, they actually have more of these kinked molecules in their membranes than us, where our membrane is a little more viscous. It doesn't flow quite as well because we don't live in super cold temperatures. Okay, so membranes self-arrange in this phospholipid bilayer, and membranes form a barrier, and they're going to let certain things across and other things not across. And we'll come back to what can go across, but we need to talk about a very basic principle called diffusion. Diffusion, and I definitely want you to know this definition, is the movement of molecules from an area of high concentration to low concentration. So if we start with this, right, we have some sort of what we call semi-permeable membrane. That means our molecules can go across it. We start with high concentration here. That's how many molecules are dissolved in a specific liquid. Let's say this is water, right? There's a unit called molarity that tells us this. I just want you to have heard that one time, but we don't really need to understand it. This is high concentration, right? Over here, there are less red molecules, so this is a low concentration. So over time, diffusion will occur because nature tries to balance the concentrations. It wants them to be equal on either side. So molecules will move from high concentration to low concentration. So over time, they diffuse across, and now the concentrations are equal. You might not have a great idea of this like in your everyday life, but I guarantee that you have seen this before. Have you ever taken like a glass of water and dropped food coloring into it? Okay, you're starting with high concentration dye right here, right in the food coloring. It drops in. It's in high concentration. The rest of the water, it's at a low concentration. So over time, the dye will diffuse out until the concentration is completely equal. This is an important principle because our membranes can have diffusion across them, but only certain things. This is when they're selectively permeable. Things that are hydrophobic, small molecules, oxygen, carbon dioxide, nitrogen, these can move or diffuse across the membrane very, very easily. Some smaller but slightly larger uncharged polar molecules, water, glycerol, these can move across the membrane, but not quite as well, but they will still diffuse. Very large molecules, uncharged things, polar molecules like sugars, like glucose and sucrose, they're too big to diffuse across. So we have to have them move in a different way, right? You have to have sugar get in and out of your cell. Otherwise, your cell's going to die. It needs energy from these sugars. Ions have a charge. They don't move across at all. we have to have a way to move them in and out of the cell. So we'll talk about how that happens in just a moment. The general rules are membranes are permeable to hydrophobic molecules. Small nonpolar, like oxygen, will pass freely through. Impermeable to charged molecules. So large charged polar molecules don't pass freely through. This is really important for pharmacology, right? If we need to get drugs into our cells, they need to be membrane permeable. A lot of them have weak acids or bases attached to them. That's really important to get things into our cells, right? If it can't move across the cell membrane, it can't get in and do its job. So pharmacists or pharmacologists think a lot about getting things across membranes. There are factors that change how fast diffusion occurs. Temperature, higher temperature tends to have faster diffusion. How soluble the molecule is in the membrane, the size and shape, as we talked about, the surface area of the membrane. We saw in the cholera example, we have all those little villi, those little finger-like things on our intestinal cell membranes. That increases the surface area. Things can diffuse from the intestines into the cells much faster. The thickness of the membrane, and of course the concentration gradient. So that means how strong is the high concentration versus the low concentration. That would be our gradient. If it's very equal, it's going to happen very slowly. But if it's like really high versus really low, it's going to happen much quicker. But how do we get other things that won't diffuse across the membrane from the outside in or the inside out? Well, this is where our transmembrane, trans means across, so transmembrane means across the membrane, transmembrane proteins can help us move larger molecules. They're basically like little tunnels that form through the membrane, and they will allow things to move through. There's some that do other jobs, but a lot of them are transporters. We have some that just operate by diffusion. We call those passive. And there are some that are like pumps. Those are active transporters. We also have a related principle. This is called osmosis. Osmosis is the movement of water across a membrane. So, as you know, some things like sugars can't move across a membrane. Water can. This causes a problem. If you have a high concentration of something on one side and low on the other, nature doesn't like that. It wants them to be balanced. So this process of osmosis actually will move the water from the low concentration area to the high. So this is a bit tricky to think about. This is the movement of water. It's a special type of diffusion dealing just with water movement. It's when whatever's dissolved can't move across the membrane. So in this example, we have a low concentration here, right? These purple things are at low concentration and high concentration over here. Nature wants to balance these concentrations. So it does that by taking water from this side and moving it to this side. And that will lead to a balanced concentration. These concentrations are now equal. So there's equal amounts of solute to solvent. Okay, so how does osmosis affect you? Well, if you've ever gotten an IV, you hope the person that made that knew something about osmosis, because our cells, right, if we have equal amounts of solutes on each side, so the red molecules are our solutes dissolved in our solvent, they can't move across the membrane, but water can. If they're equal, we call this isotonic. The solute concentration is equal inside and outside the cell. If we have a scenario where, let's say, we have less solute outside than inside, right? Less concentration outside than inside. That is what we call a hypotonic. Hypo means low. So it's a low concentration outside solution. This would be like if you injected pure water into someone's veins, right? There's lots of dissolved sugars and things inside of your cells. Pure water would be hypotonic. This would cause osmosis, and the water would move into the cell trying to lower the concentration, and your cell would start to expand and expand until suddenly it can't expand anymore and it bursts and explodes. Now, you probably can guess that cells exploding is not a great thing if you want to stay alive. So this is why there is saline added to IV solutions, right? It helps balance the what we call tonicity, right? So in the other case, if we had too much salt in an IV solution, it would create a hypertonic solution. So hyper means high, and tonic, right? That's dealing with the solutes. So there's lots of solute outside, lower concentration inside. We're going to try to balance this by water moving out of the cell. That's going to cause your cell to shrivel and shrink. And that's just as bad as, well, it's almost as bad as bursting, right? It's not going to function properly when it's all shriveled like this. So hypertonic solute concentration is greater outside the membrane than inside. Hypotonic solution is greater inside the membrane than outside. And that's going to cause water to move. Water is going to move from the low concentration to the high concentration. So it's kind of backwards from diffusion, right? And we saw an example of this happening in our previous example at the beginning of the chapter with the cholera toxin, right? Cholera toxin pumped these ions outside the cell, making a hypertonic solution. That caused the water from the intestinal cells to rush outside, shriveling the cells and leaving us dehydrated, and then causing massive diarrhea. So this chemical principle is very critical to understand. That's why with the oral rehydration therapy, we feed them sugars and salts to get that tonicity back to equal to isotonic so that water can actually go into the cell. So here's an example with red blood cells like I talked about, right? If they're in a hypertonic solution, water gets sucked out because there's lots of solute outside, they shrivel up. If they're in isotonic solution, it's equal, so they stay nice and plump and properly shaped. Hypotonic solution, there's not enough solute outside. Water is going to go inside to where there's a high concentration, and it's going to cause the cells to swell and eventually burst. So if you don't have your IV fluids properly mixed, right, you could seriously injure someone by injecting pure water into them. This example might help you think about that a little bit clearer. Okay, so we talked about hypo and hypertonic solutions and isotonic solutions. I'm sure the only tonic solution you want to hear about right now is a gin and tonic. Am I right? That's my one tonic joke. I got to get it out of the way.
Just as solutes tend to diffuse from regions of high concentration to regions of low concentration, so do water molecules. Osmosis is the diffusion of water across a selectively permeable membrane from regions of high water concentration to regions of low water concentration. The concentration of water in a region depends on the amount of dissolved solutes in the water. The water concentration is low when the concentration of dissolved solutes is high. Conversely, the concentration of water is high when the concentration of its dissolved solutes is low. A lipid bilayer is a selectively permeable membrane. Lipid bilayers allow water molecules to pass through, but block many dissolved solutes. Here the solutes are blocked from diffusing down their concentration gradient across the membrane, but the water molecules can cross. Osmosis across the cell membrane must be tightly controlled to preserve the form of the cell. A higher solute concentration outside the cell is called hyperosmotic or hypertonic. When the solution outside is hyperosmotic, osmosis causes a net loss of water from a cell. The cell shrinks and the cell solute concentration increases. A lower solute concentration outside the cell is called hypoosmotic or hypotonic. When the solution outside is hypotonic, osmosis causes a net uptake of water. The cell swells with water and the cell components are diluted. If enough water enters, the cell lyses. The cell membrane ruptures and disperses cell contents. When a cell is in an isotonic or isoosmotic solution, in which equal concentrations of dissolved solutes are inside the cell and out, the two are in osmotic balance and water will enter and exit the cell at equal rates. That is, no net movement of water occurs. Free-living cells, such as microbes that do not live in isotonic environments, have a variety of adaptations for survival. For instance, the ocean-dwelling bacterium, Vibrio cholerae, lives in a high-solute environment. These bacteria accumulate solutes known as osmolytes to ensure that they are isotonic to the external environment, thus preventing water loss. Other microbes live in hypotonic environments and possess a rigid cell wall external to the cell membrane. As water enters by osmosis and pushes against the cell wall, the wall resists the tension and pushes back with an equal but opposite force, called turgor pressure. Turgor prevents water movement and the cell does not lyse.
Okay, so think back to the cholera example as it mentioned. We have the toxin goes in, the toxin turns on pumps. These pumps are going to pump out ions, right? These ions make a hypertonic solution in the intestinal lumen. That hypertonic solution draws out water because it's trying to balance the concentration. It draws out the water, causing the cells to shrivel and dehydration to happen. That leads to diarrhea. And if you don't replace the ions with sugars and salts inside the cell, you're not going to be able to get water to go back in. So that's why we use that oral rehydration therapy. So I think this ties together a lot of principles here, right? We've talked now about membranes and how they're constructed. We've talked about ions. We've talked about concentrations and osmosis. So I definitely would like you to understand what's going on in this example for our exam. Okay, to recap 4.5, a membrane consists of lipids and proteins. There's some other small molecules in there. The phospholipid bilayer has those two faces, right? The membrane's fluid, it can move together. It's not actually tightly bound together. Transmembrane proteins can go across the membrane. Membranes are only selectively permeable. Only small hydrophobic molecules can diffuse across the membrane. Other things can't do that, so we need those transporter proteins that can move molecules across the membrane. And then osmosis is the diffusion of water across a membrane from a region of low solute concentration to a region of high solute concentration. Make sure you also know the definition of diffusion as well. Okay, that's it for this section.